Hello, my name is Carl Schiesser, and I'm the Director of the ARC Centre of Excellence for Free Radical Chemistry and Biotechnology. Now, you're probably seeing this video because you've come to the Concept Radical website and are wanting some more information about free radicals, and that's what I'm here to tell you about, some basic concepts and basic facts about free radical chemistry. Now, free radicals are everywhere. They cause aging-related illnesses in people, they deteriorate plastics, they fade houses in paints and works of art, and they can contribute to heart attacks, stroke and cancers. What you will have seen in the media and in advertising in newspapers and magazines is advice about antioxidants. And we all know about antioxidants. Antioxidants are out there to combat free radical damage. In this example, you can see that free radicals are depicted as a nasty green critter with teeth and horns, and if you were to buy this particular brand of antioxidant, complete with a free halo, it combats the free radical damage from this green nasty critter who happens to be surfing a red blood cell in this advertisement. Now, free radicals are not green nasty critters with horns. They are molecules with unpaired electrons, and I'll talk about that in a little moment. Free radicals appear in all sorts of advertising, cosmetics. There are slogans like "Stop the visible signs of aging." We have Dilmah tea, which tells you to drink at least 2 cups of tea a day in order to get your recommended daily allowance of antioxidants. There are breakfast cereals with antioxidants. There are drinks with antioxidants. And of course, red wine contains antioxidants. It's recommended that if you were to drink as an adult male 2 glasses of wine a day, 2 glasses of red wine a day, you'd get your necessary daily allowance of antioxidants. White wine does not have the same level of antioxidants. So what are free radicals? Well, free radicals are molecules that have unpaired electrons. The question that many of you are probably asking yourselves is, how are free radicals actually generated? And I'm going to actually demonstrate how free radicals are generated through the use of my trusty assistant over here, Stephanie. So, as many of you will be aware, bonds in molecules are formed by the pairing of electrons, just like a handshake. If we consider that my hand is 1 electron and Stephanie's hand is 1 electron, then we can form a chemical bond through the pairing of those electrons. Electrons like to be in pairs. That's a quantum mechanical principle. As a result of forming the bond, everybody's happy. Occasionally, things like ultraviolet radiation, pollution, biological free radicals can come along and cause this bond to break. And when the bond is broken, I become a free radical because I have an unpaired electron. Stephanie becomes a free radical because she has an unpaired electron. And because we're reactive, we wish to pair up that electron with something else, and we go about doing that through reactions with biological molecules, if you happen to be generated in a living system, or perhaps we attack the polymer matrix of a paint, or we might attack a work of art, simply to satisfy that unpaired electron. Thanks, Stephanie. Now, out of a quirk of nature, molecular oxygen also is a free radical. It's in fact a diradical. And that has to do with, again, with a different quantum mechanical principle. And because molecular oxygen is a free radical, it can become involved in reactions that we call oxidative stress, can cause oxidative stress, and can generate other reactive oxygen species. So in this particular example here, I have molecular oxygen reacting with a hydrogen atom in some molecule. Let's pretend it's a biomolecule. By satisfying itself, it generates a free radical in that biomolecule, which can go on to react and produce other potentially damaging free radicals. If that free radical is generated in DNA, you can lead to some serious problems from a health perspective. So the message is oxygen is toxic, but we need oxygen to live. The free radical nature of oxygen allows it to bind to hemoglobin and become transported essentially as energy in most living things. And the other take-home message is that antioxidants protect against free radical damage. But in most healthy diets, there are plenty of antioxidants there, you don't really need to pop extra antioxidant supplements, but perhaps if you have a high exercise regime, you may wish to supplement your natural antioxidants with a few extra ones. So free radicals are caught up in a whole bunch of diseases. There are a number of important diseases that have free radicals associated with them, but I want you to focus for just a moment on some of the top-selling drugs in US dollars, global sales in 2008, and what they are there for. So the top-selling drug is Lipitor, and that's a drug for high cholesterol. Nexium is actually the number 2 selling drug at $4.8 billion in 2008. It's for heartburn. But then Plavix is supposed to be a remedy for stroke or heart attack. Then you have diabetes medication, and then you have hypertension. And heart attack and stroke, diabetes and hypertension all have free radicals associated with those diseases. There's another childhood neurodegenerative disease called ataxia telangiectasia, which is an inherited disease, and it's also a disease which has free radicals associated with it. One of the things that most scientists don't really understand very well is how free radicals are associated with these diseases. And in order to understand how free radicals are associated with these diseases, it would be really good if we could somehow visualize them in living systems. In this next slide, I'm going to show you some of the technology that has been developed recently through members in the ARC Centre of Excellence for Free Radical Chemistry and Biotechnology, which is applicable to a broad range of free radical mediated problems. So for example, in the polymer industry, if you have a polymer coating, be it in a coronary stent or perhaps in an orthopedic implant, you don't want those things to become affected by reactive oxygen species and free radicals that might be floating around in your bodies, because the last thing somebody wants is another operation a few years down the track to replace either a stent or an orthopedic implant. And so we have to make sure that the polymer coatings on these types of materials are robust and can withstand the oxidative stress of the environment that they are in. Also in your car, you like to, when you buy a new car, you'd like it to remain shiny forever. But of course you drive out in the sun and in the rain, in the pollution, and all those factors can affect the performance of the polymer coating on your car, and as you all know, the cars become dull. And there are a whole bunch of other components for example, in computers and in other types of technology that also suffer the same sort of fate. So here's an example of a car which has had obviously been involved in an accident. It's had a bad spray paint job that has fixed the paint to begin with. But as you can see, if you zoom in on the paint of the bonnet, you can see that there's always cracking and peeling that's come about in a period of about 2 years, which is the result of a poor quality paint being applied to that surface and simply weathering, ultraviolet radiation, etc., damaging that paint. So in our centre we have, through talented researchers, Catherine Fairfield-Smith and Steve Bottle in Queensland, developed what are called profluorescent nitroxide that glow in the presence of oxidative stress, and they glow in the presence of free radicals. So you can get a direct measure of where free radicals are and the quantity of free radicals that are actually involved in the degradation of a whole bunch of materials. And so in the right-hand panel over there, you can see a piece of polypropylene which has had the profluorescent nitroxide embedded in it. And you can see that after 16 hours, 24 hours, 40 hours and 48 hours, at elevated temperature and in air, it starts to glow. And that glow is telling you that the polymer matrix is degrading, and it's degrading through oxidative stress and through the generation of free radicals. And it's this sort of technology that you might be able to incorporate into important components in a variety of industries to actually give you a signal to tell you that the component, for example, if it is a component, is degrading and that you need to do something about it. Those same fluorescent nitroxides, or very similar I should say fluorescent nitroxides, can also be used in biology. And so we've also been working at making fluorescent compounds that can detect free radicals in hypertension-related diseases. And in the panel up there you can see a small amount of that compound glowing in solution. And when we incorporate them into cells, and unfortunately this doesn't show it very well, you can see that if we incorporate them in cells that are related to hypertension, the fluorescent compounds get introduced and incorporated into the cells and they start to fluoresce. And that tells you that the disease is prevalent. Now, as I mentioned right at the beginning, free radicals also are responsible for the degradation of works of art, either through pollution, through the involvement of radiation, simply through oxygen reacting at weak points in, for example, paint. And for example, if it was an outdoor mural, they're very, very susceptible to ultraviolet damage and oxidative stress. So art galleries, in particular, they like to have very climate-controlled conditions to minimise that oxidative stress. But we still don't fully understand how the media that are used in, for example, modern works of art perform in relation to free radical damage and oxidative stress. Free radicals and oxidative stress are also involved in the formation of biofilms on important works of art, and they can be controlled simply through the use of nitric oxide, which is a very simple free radical that is involved in cell signaling. And so you can see that in cultural materials, an understanding of how free radicals work and how they function in terms of the biology and the chemistry of the cultural materials that we're looking at can have an important impact on our understanding of the chemistry and our ability to control and treat works of art that have been damaged through free radical mediated processes. And so I'll just leave you with an important image here, Blue Poles number 11 by Jackson Pollock, reputedly worth some $200 million. If you have a $200 million work of art in your collection, you want to make sure that it is housed well, that you understand the performance of the components in that particular work of art, and as you can see, it is in fact a very, very important priority for galleries and in fact for the government, because Blue Poles number 11 is owned by the Australian government. It's important to be able to understand what's going on and to support research that allows us to better conserve and better treat our important cultural heritage. So, thank you for listening, and I hope that this presentation has provided you with some background into free radicals and how they impact in biology and in materials, in particular relating to cultural materials. Thank you.